So let's just go over biology paper 2. This is the 2020 internal exam. So beginning the first question here, figure shows a plant cell and we want to name the parts which have been labeled A, B and C. So if you look at A is showing this big space inside the cell. What do we call that big space on the middle of the cell? That is known as the vacuole. And then B is showing the biggest, you can see this structure, the biggest organelle that is on the side, call it the peripheral of the cell. That is the nucleus. And then C, you can see it is pointing to the layer that is inside the cell. Inside, not the outermost layer. The outermost layer is the cell, but then this is the cell membrane because it is outside. We can as well say it is the plasma membrane. So these are the parts here. And then the next one, name the organ in the plant in which this figure is found. So if you see such a cell like this one which goes up down, that is known as a palisade cell. And it is found in which organ? It is found in the leaf of the plant. Why are we saying so? Because we know that palisade cells have got a multitude of chloroplasts, and these are the ones that you can see. All these dots, those are chloroplasts which will contain chlorophyll, which is a green pigment of the plant. Describe two adaptations of the cell in the figure which enables it to carry out the function. Why is it that a palisade cell, what is the function of the palisade cell anyway? The palisade cell is involved in photosynthesis. One reason for that, it is because it contains a lot of chloroplast. So how should you write this? You are going to say abundant or a lot of chloroplast. And this contains chlorophyll, a green pigment that is going to be able to carry out photosynthesis. Another reason is that the cells are actually arranged. Look at how they are up down. So they are vertically arranged, and that concentrates the chloroplast to one end of the cell, right? So that is another adaptation, just the way the cells themselves are arranged. So you can say the cells are longer vertically than horizontally. That allows the chloroplast to migrate up and down when there is a change in light intensity. Explain what would happen to the cell above if it was placed in a test tube containing distilled water. Now you know that the cell is highly concentrated. It is salty inside because it contains a lot of things inside. What happens is that when you get this cell, you place it in distilled water. By diffusion, you know that water is going to move from a region of high water potential to a region of low water potential. So the water is going to be going inside the cell to dilute the cell. And so the cell will become big. But because it's a plant cell does not burst. So it will undergo what is known as a turgidity. Turgidity is where the cell becomes big because it has gained water. So you just need to explain this turgidity to get the other one mark. The cell is going to gain water because water moves from a region of high water potential to a region of low water potential. And the cell becomes big. That is known as turgidity, right? And then we have got this question here. The figure below shows the internal structure of a tooth. And the first one is we want to label or to name the parts D and E. What are these parts anyway? We can label everything even starting from the top. You can see the outermost, this white thing of the tooth that you can see, that is known as the enamel. So F is the enamel. What you can see inside here, D, these are actually nerves. So this is a nerve. And then E is showing this structure, the bony structure, the hard part of the tooth. This is called the dentin. So these are the parts that we are able to see here. This structure here, though not labeled, that is the gum, right? So these are some parts that we should know. Explain what happens to the part labeled F during tooth decay. So what will happen to the enamel during tooth decay is that it's going to be destroyed. So this thing is going to be eaten up, and then you are going to have a space inside there, a hollow space. So it's going to be eaten up in tooth decay. Okay. Looking at the shape of the tooth suggests the role it plays in digestion. So look at this. What type of teeth do you think this is? So this can be the molars, the big teeth, and these are going to be involved in crushing things. Crushing. Something that is used for tearing is going to be much sharp. Complete the human dental formula that you can see here. We have got a total of 32 teeth, of which the incisors, we have got 2 on top, 2 down. How about the canines? 1 on top, how many are going to be down? And then the premolars, we have got 2 on top, 2 down. And then the molars. Normally for a human dentition, on top and down, they should be the same number. So if on top we have got 1, down we're also supposed to have 1, on top 3, down also 3. So let's count them, and there are 2 plus 2, that is 4, 1 plus 1, that is a 2, this is a 4, this is a 6. So 4 plus 6, that is a 10, 2 plus 4, that is 16. Okay, I mean 2 plus 4, that is 6, 10 plus 6, that is 16. So you multiply this number by 2 because we're only taking 1/2 of the mouth. That will give you a 32. Okay. So what was G? G is supposed to be a 2. Why? Because we multiply this number of the teeth that we find here by 2 for us to be able to get FH2. And then H is what? 1. And then J is 3. So that is it. And then question 3. The figure shows a section through a nephron. Nephron is a functional unit of a kidney that makes urine, and we want to name the parts J, K, and L. You can see J is these capillaries which are in this thing. So this thing that you can see is known as the Bowman's capsule. Bowman's capsule. And then these tough capillaries that we can see, that's known as the glomerulus. Glomer, this is RULUS. Okay, so I've already labeled what K is. K is the Bowman's capsule. What other thing was supposed to be labeled? So you can also see this is a collecting duct, right? It's collecting the urine from all these different nephrons, and that is going to go to the ureter, which is going to go to the bladder, and then it is going to go to the urethra. All right. So I can label for you other things. You can see these convolutions that you can see at this point near the Bowman's capsule. This is known as the proximal convoluted tubule. You also have these ones which are away, these convolutions that are away from the Bowman's capsule. This is known as a distal. Distal means far. Proximal means near. So this is distal convoluted tubule. And then this, which is draining into the collecting duct. So you also have this part that is draining into the collecting duct. That will be the collecting tubule. And then you have got this artery or artery that is bringing blood to the glomerulus. That is the afferent arteriole. Afferent because it is towards. It is a small artery, so it is known as arterial. And then this one that is taking blood away is known as efferent, arterial. Okay. And then you also have these arteries which you can see here. These are known as the peritubular capillaries or arteries, arterioles actually. Suggest the three substances that are not filtered out at J. So what three things should not come out of blood to go into the urine normally? Okay. Can you name anything? So one thing is that the red blood cells should not be filtered. The reason for that is you don't want blood to go into urine because that is indicative of a condition. All right. What other thing should not be filtered? Also, the white blood cells should not be filtered. They're supposed to remain in blood. And also, big proteins are not supposed to be filtered. So, red blood cells, white blood cells, and also big proteins are not supposed to be filtered. Explain what would happen at J if the coiling of the blood capillary was increased. So if the coiling of the blood capillary, which is the glomerulus, was increased, it means that there will be actually more time, because when blood is going like this, it is going to take more time for the blood to leave. So blood is going to spend a little more time here to be filtered. And so that is what would happen. Blood would spend more time in the Bowman's capsule, and that will increase time of it being filtered. Name the hormone that causes resorption of water in the kidney and the organ where it is produced. So there is an hormone that is going to come and act on the distal convoluted tubule, and that is known as the. Okay, I wanted to mention something else. So first, one thing that we need to know is that when we have got, imagine it is very cold. You don't want water to be lost in urine. Why? Okay, when it is very cold, you are not let me say start with when it is very hot. When it is very hot, what I just mentioned is true. When it is very hot, you are losing water via sweat. So that is why you see that when it is hot, you barely go to the toilet to urinate, because most of the water is being lost via sweat and not urine. But opposite, when it is cold, you are going to have water being lost through urine. So if you drink a lot of water and it is cold, you'll be mostly urinating. That is because you are not sweating, and so that is the only way you are going to be losing water. Okay. So what will happen is that when we do not want to lose a lot of water, and a hormone is going to be released from the. If you look at the kidney, the way kidney is like this. On top of the kidney, we are going to have a gland there. But to be specific, we have got what we call the adrenal gland. So the adrenal gland is going to be able to produce one hormone. I don't know why this one is coming well, so let me mention it because it's also part of this topic. There is an hormone known as aldosterone. This hormone is going to be necessary in the reabsorption of salt. So imagine maybe a lot of salt was taken into blood. I mean into this structure. Because normally you don't need a lot of salt to move out of blood to go into urine. So if it was taken, salt is going to go back away from this tube that is forming blood to go, that is forming urine, to go back into blood. The hormone responsible for salt reabsorption is aldosterone. Now, when you want to reabsorb water, the hormone that is going to be produced is the antidiuretic hormone, known as ADH. and diuretic hormone. And this one is produced by the hypothalamus, right? So that is what happens. Name the organ that causes the resorption of water. So we have named, and also the organ is the hypothalamus. And then we have got this one. A figure below shows the external parts of the human eye. Name the parts labeled M and N. So this M, this is space that we see on the middle of the eye that is known as the pupil. And then we have got this whitish thing that we can see here, that is the cornea. It is a continuation from the behind of what is known as the sclera, right? And then you also have O. What is O? So this is known as the iris. And the iris contains two types of muscles. It has got longitudinal muscles and it has got circular muscles. If you want to be more fancy, muscles that go like this, these are the ones which we call longitudinal, because they pull the pupil to make it bigger, so they are also known as dilator pupillae. And then you have got circular muscles like this. These ones, when they contract, they'll make the pupil to become small, all right? The constrictor pupillae. So we have labeled the parts M and N. Explain how N is adapted to its function. So N is the cornea, and what happens that when I can draw the eye in this view. So this is the cornea like this. Then you are going to have the space like that, which is going to leave the pupil. So when light is coming like that, the light is going to be turned such that it goes and passes and then it hits the back, and that is going to cause, what is it going to cause? Is going to cause an image to be formed, and that image is what actually enables us to see. So you are able to see things because the cornea contains, it's a lens, and that lens enables us to see. Okay, so that is what we have to know. So sometimes the cornea can actually make the image to be formed much earlier than behind where you are seeing I've drawn. And sometimes the meeting point of those two lines that have drawn will be much far away, and that is called long-sightedness. If it is near, that is known as a short-sightedness. Okay, so that is what happens. So it is adapted to its function. It is made up of a lens, and that enables the converging of the rays when we get to see, and that is going to help us to be able to see. Okay, what is the function? The cornea is transparent, like you can see through. It's a transparent section of the sclera. The sclera is the entire structure, the entire thing that this thing here. This is the sclera. But if you come in front, that is known as the cornea, which is transparent, and it will do refraction, the bending of light. Okay, and that is going to lead to the formation of an image. Describe the role played by M and O when a person moves to a room with a dim light. So if you go to a room that is dark, what is supposed to happen is that the pupil is supposed to become big. But what is the role of M and O? So for M, the pupil is going to be dilated. It is going to become big. How about O? We've said that O is the iris, and what will happen is you need to explain what happens to the longitudinal muscles and to the circular muscles. So the longitudinal muscles will be pulling in this direction. Okay? When they pull in this direction, what will happen is that the pupil is becoming big. So the longitudinal muscles will contract, but the circular muscles will relax. In the other way, if you go in a light, in a room that has got a big lighting, the pupil is supposed to become small. So what will happen is that the longitudinal muscles relax, the circular muscles will contract to make it small. That is why you can see this question is a 3 marks, because when you're explaining about the iris, you need to explain the longitudinal muscles and also these circular muscles. M, which is the people just say to become big, to dilate, right? Explain the cause of short-sightedness and how it can be corrected. Now, when you look at the eye, the way we drew it like this, in short-sightedness, what will happen is that when light is coming, it is going to converge. So you can see things which are near, but you can't see things that are far. Imagine when you are looking at something that is far. So when light comes, it's supposed to be refracted. So when it is refracted, it's going to bend and it's going to merge area. You can see what happens. So this is where the the rays of light has merged. It's supposed to merge at the end here, but if you are looking at something that is near, what would have happened if you are looking at something that is near is light is going to be going like this, and then it's going to bend, and then also it is going like that, and then it's going to be able to bend. You can see the image is formed at the end on the sclera here, or the retina. So you'll be able to see if something is near, but not when something is far. So the way you correct this is you can use a concave lens. And for a concave lens, what happens? A concave lens is actually going to just bring the object, it's going to reduce the refraction of light. A concave lens looks like this. A convex looks like this. So what will happen is that when light is coming like this, it will not be turned area because this lens makes light to go like this. But this one makes light to go like this to meet. Okay, it's convex. This one is concave. So because of this, light is going to be a little bit turned in this direction, right? Such that by the time it will be reaching here, then it is going to have a bigger distance, and then it will come and merge at the end of the eye there. If this wasn't here, light wouldn't have been turned like a bit like that, and so it would have merged somewhere earlier before reaching the end. So that is how you can actually get to treat that. Okay, so how do you treat short-sightedness? You can use what? A concave lens. That is very important, right? And then the last question on the section A. Red green color blindness is a sex linked characteristic. Define sex linked characteristic. A sex linked characteristic is a characteristic that is passed on a sex chromosome. A characteristic passed on the sex chromosome. We have got two sex chromosomes, the X and Y. If a person has XY, then they are male. If someone has got XX, then they are female. What can cause X-linked genetic disorders like the red-green color blindness? Okay, there are a lot of different things that can cause X-linked characteristics. They can actually be passed. Okay, they can be passed just from parents to offsprings like that, and so you end up with such a characteristic. So in other words, they're inherited. Okay, they are inherited. Okay, a normal couple had a colorblind son. Using the gene R for normal color vision and gene small r for color blindness, write down the genotype of the father. So this color blindness is actually recessive. Okay, the meaning of that is when you have got, like for the father, the, I've said that males have got X, Y, right? Now, the Y chromosome never carries any sex linked characteristics, only the X chromosome. So, because we have got a normal couple, meaning that the father, because when you have got capital R, then you are what? You are normal. So this is going to be capital R, normal. I've said that Y cannot carry the gene for being sick, or the gene which is showing the color blindness. For a mother, what have we been told? A normal couple of had a color blind son. So if both the father and the mother are normal, but the son is sick, the only thing means that the mother, even though she is normal, she's a carrier, meaning that she had one R capital and then she had the small r. Okay. A father cannot be a carrier. It's either you are normal or you are sick. But a mother can be normal, but then she's a carrier. Using the genetic diagram, show how the son could have been born colorblind. So the first thing that you need to write is the phenotype. What is phenotype? Phenotype is a physical characteristic. Physical. What you can see. So I've got a normal man. We are crossing with a normal woman. That is a phenotype. And then now we write the genotype. What is the genetic makeup? Genotype. Genetic makeup. You can see the man is X capital R, Y. We are crossing this with X capital R, X small r. The next thing that you write every time you are writing this diagram, a genetic diagram, now you write a gamete. So gamete, what is going to be found in the sperm or in the egg? So for the gamete, you just get these same ones, X, R. Now you are separating them, put it in a circle, and then Y, also put it in a circle. Don't put a times here. And then X, R, put it in a circle, and then X, capital R, small r, put it also in a circle. So these are the things that you find either in the egg or in the sperm. And then fertilization is going to happen. So during fertilization, this can go with this. So that will be X capital R, X capital R, or this can go with this. That is going to be X capital R, X small r. This can go with this. You always start with X capital R and then Y, right? And then this can go with this. That will be X small r and then Y. So these ones you can see, because you can see these are letters. This is a genotype and this is the offspring. We call it as F1 offspring. So it is F1 genotype. The F1 genotype, you underline it. Okay. And then what will be now the F1 phenotype? Phenotype is a physical characteristic. What will be the physical characteristic? When you have got XX, that means it's a male. But both are, sorry, it's a female. Both are capital letters. So this is a normal female or the normal daughter. How about this one? She has a small r there. She's still normal, but she's a carrier. You can say normal female or you can say carrier female, but she's still normal. And then this male, XY, is a male, but there's a capital R. So it is a normal male. And then you can see this one has a small r. For a male, it's either you are normal or you are sick. So this one is a color blind male. So you can see there are 4 physical characteristics. Of all these 4, there is only a chance of 1 out of 4 being sick. 1 out of 4, if you convert it as a percentage times 100%, that means that there's 25% that a male child that is born is going to be color blind. So that is how you actually get to go about these questions. You can as well go to the essays. You choose which essay you can write. Describe the health risk associated with fetal development. Describe the benefits of possible risk of using and possible risk of using contraceptives. Explain other methods of birth control in human apart from the use of contraceptive pills and injections. So you can as well get to look at these. So they are really interesting essays that you can pick up and get to write. Just make sure that you go through the notes and you understand what are specific things that you'll be able to talk about, and we'll be of course talking about in later videos how to best answer the essay part, right?